Well, it's very close to 10 o'clock. I've haven't prepared a detailed CV for Dr. Gretchen Gerrish, but I know that she is the director of the Trout Lake Research Station in the Trout Lake, North Central Wisconsin, and has been for some time, has graduate students there and runs a program. And I've heard about your work, Dr. Gerrish, with regard to a bioluminescent organism, I think written up in Science. And so I was hoping that even though our topic is Wisconsin and fresh water in Wisconsin, that you might tantalize us with some of your marine research as well. I look forward to your talk. So you now have the floor.
Thank you, Richard, and thanks for the invitation. It's always fun to hear about different groups that gather around topics, especially related to climate change and some of these big ideas that our world is handling. I will give just a little bit of background on how I ended up here to kind of orient you to where how my feet got wet in the field. And I didn't add a lot of marine work to this talk today because I wanted to highlight some of the climate work in Trout Lake Station.
Sure.
But I'm thrilled to talk about it. I just got back from the field from a marine trip, so that work proceeds as well. So hello, I'm Gretchen Gerrish. I am a limnologist. I consider myself an evolutionary ecologist who really engages with all the microscopic creatures living in our freshwaters and marine systems and how evolutionary and ecological times interact. Those are kind of the broad questions that I tackle in my research. I started out with at Lawrence University for my undergraduate, where I studied biology. And during my time as a student there, I actually got the opportunity to spend 2 summers conducting lake research at Trout Lake under the guidance of the then director, Tom Frost, and Bart DeStasio, who's from Lawrence University. So my connection to the Center for Limnology in Trout Lake Station started early in my career. But after that time, I ventured far afield. I did my master's at the University of Illinois in Champaign-Urbana, and then finished my PhD at Cornell University, where I did mostly marine research during that time. I'm a Midwesterner, so I had the opportunity to come back. I worked at Notre Dame, running a field program for 3 years during my postdoctoral work, and then was a professor at the University of Wisconsin-La Crosse for 9 years before joining Trout Lake Station in 2019. So I'm relatively new to my position here. And it was interesting because I started at Trout Lake about 6 months before the pandemic hit. So my first 2 years of the job, I had all this vision and opportunity and got to tell everybody no for 2 years is basically. But I'm excited. We had a full summer this last summer. So we've been back open completely and had about 35 graduate, postdoctoral, and undergraduate researchers on site conducting projects. Today, what I thought I'd start with is just a little overview for those of you that aren't familiar with Trout Lake Station and its place in the broader field of limnology, and then dive into a project that I'm collaborating on relating to our shifting understanding of ice dynamics and how that impacts the phenology of lakes and lake creatures. So that's where we're headed today. And really what we do is about water. And so I like to center a lot of my talks just thinking about water in general. The idea that the globe really is covered in water, and most of it is salty. So 71% of the surface is covered by liquid water. But when we ball that all up, that surface, all of the water fits in the big marble shown here. And then our freshwater resources are equivalent to the small circle that you see here. And then that tiny little dot is all the fresh liquid surface water in proportion to the size of our planet. It's incredible to think about the Great Lakes region in the state of Wisconsin in that context, because the Great Lakes contain about 21% of the Earth's fresh surface liquid water. And in that, with Wisconsin nestled right here within the Great Lakes region, we have a huge responsibility and a huge wealth that really contributes to us being nestled as the heart of limnology and the study of lakes and freshwater resources. So Wisconsin, the estimates or the current numbers are 32,000 miles of river, 5.3 million acres of wetlands, and 15,000 lakes. Those numbers are incredible. I know Minnesota claims to be the land of lakes, but Wisconsin really has it beat if you actually compare the numbers. It depends on how you define the water bodies, but really Wisconsin is a heart for the understanding of lakes. And of course, these lakes were formed by the retreat of the Laurentian ice sheet. So up here in Wisconsin, especially northern Wisconsin, I live in Vilas County, and Trout Lake Station is located in Vilas County. The lakes here were formed during the retreat of the Laurentide ice sheet. And as that ice sheet dug back, it basically scoured out what we call kettles, where water was retained or large pieces were dragged through and scoured out these deep openings where water pooled and have been sustained and formed the rich lake environments and connections between them that we have today. So in that context, many of the lakes in this region are only 10,000 to 15,000 years in age as that ice sheet retracted. But here's a picture of Vilas County. And sometimes people refer to it as a landscape full of water. I like to refer to it as a waterscape with some land, because over 50% of the area that you see here is either lake, wetland, stream, or river, with waters coming very near to the surface. And so just for orientation, if you go down this road right here, you could go all the way down to Madison. This is Highway 51. This is Trout Lake. It's one of the largest lakes in the region, and our research center is located here just on the south shore. But each one of these small lakes and its structure, the characteristics, the chemical characteristics within those lakes are very dynamic and are dependent on how high the lakes are in the watershed. Two of my favorite examples are some of our long-term research lakes, and that's Crystal Lake and Big Muskie Lake, these two. Crystal Lake is high up in the water table. All of the water that comes in there comes from rainwater, and it's called crystal because it's crystal clear, and the drainage is a sandy basin, very little nutrients. That lake is about 200 feet from the shores of Big Muskie Lake, but Big Muskie is almost 4 feet lower in the landscape, and it's one of those great fishing resources, tons of macrophytes, high nutrients. And if you saw the two lakes separated from each other, you would never think that they were so close in the watershed. So elevation plays a huge part in what we find in our lakes. So does the landscape around each water body. If it has a lot of deciduous trees, you can have nutrients and color to the lakes. If it's surrounded by pine trees, it can be acidic and have less growth. And so when you think of studying lakes, Vilas County and Oneida County, which contain almost 2,500 lakes of different structures and shared watersheds, it is a place where we can develop a depth of understanding while not having to travel quite so far to get to explore some of these different systems. And the Center for Limnology is one of the premier groups that works on lake research. Actually, the mission for our group is that we're really focused as an outward research center that integrates cutting edge research, training, and outreach to address the most pressing questions and societal issues related to fresh water. So I start with water because it's at the core of our goals. That's one of our mottos. We're the lakes people. And I don't know where all the crowd comes from. Are most of you guys from Madison, or is it from all over the state and beyond?
I would say most are from Madison. I'm looking down the list here from people outside. There is one from near Milwaukee and one from Gray Lakes, Illinois.
I grew up on Pewaukee Lake.
Right, yeah.
Okay. Yeah, so yeah, it's funny because that's how we identify up here too is, oh, which lake are you near, right? It's that those are our landmarks, so that's always fun to hear.
It's
So
just, if I could say something, yeah, I'm in Grays Lake, but I've spent a summer up in Minocqua, so I know that area pretty well. And what you're talking about. Thank you.
yeah, it's a stone's throw from one lake to the next. And when people ask me where I live on Little Spider Lake. I don't even mention my town. I'm on Little Spider. It's it's an identifier up here when we talk.
Alan here. Although my main home is in Rockford, Illinois, I have spent over 30 years sailing on Lake Mendota and have even borrowed the Hassler Lab dock from time to time.
So this picture is familiar to you?
Oh, very.
Yeah. So so the Center for Limnology is comprised of two facilities. We like to say we're a center with two field laboratories, the Hassler Lab, which you just mentioned, which is down on the shores of Lake Mendota, and then the Trout Lake Station Lab, which is where I'm at. Our group is composed of 12 PIs, 6 of which are faculty and 6 of which are research PIs, so pretty straightforward research. And then within our community, we have almost 40 graduate students and postdoctoral researchers, and in addition to a number of technicians. And so they're distributed between the Hassler Lab and Trout Lake. Up at Trout Lake Station, we have 10 full-time operations staff and researchers that are here all winter and all summer. And then down in Hassler Lab, they do more teaching, and a lot of the graduate students go back and forth between the two locations. At Trout Lake, I'll talk mostly about Trout Lake because that's where my work is centered, and because some people are more familiar with our Hassler Lab, so I thought I'd take the opportunity to talk more about Trout Lake. It's actually, for the UW system, serves a region that's really utilized and underserved by the UW system in general. There are some other field stations for UW Madison and Stevens Point that are up in our area. But if you look at the distribution of the UW campuses, there's a large part of our northern state where the UW and connections to the public and recruitment of students occurs through places like Trout Lake Station, the Kemp Forestry Station, or the UW Extension programs. And so we like to consider ourselves as centrally isolated in the Northwoods, about 3 hours from any of the big major airports. The history of limnology is tightly tied with UW Madison. And so it is considered one of the birthplaces, or the American birthplace, for limnology in the field of that study. A lot of that can be attributed to Edward Burge and Chauncey Juday. And they were very active in the field from 1875 all the way up to 1944. The Juday family continues to be involved with the station, both as researchers for a period and now as part of our outreach and support group. And here's just some pictures of Burge and Juday up in the corner and some of the early instrumentation that they put into effect. So many of you who have ties to Madison have probably heard of Burge Hall, which is the center for biology and zoology on campus. And I have affection for Burge's research critter. We share interest. Both of us began our careers working on a small crustacean called Daphnia, or water flea. And he did crustacean systematics. He named the group, saw that there were different kinds of this little crustacean that is a dominant one in many of our lake systems. And grew his career with a real appreciation for the ideas of evolution and the diversification of organisms moving throughout the landscape. And while his research focused on zooplankton, his career took him in many directions. And I had to write down a few notes, because when I say he had a lot on his plate, he was one of the lead researchers developing the field of limnology, but quickly moved into starting the Department of Zoology for UW Madison. He served as the Dean of the College of Letters and Science. He served on the State Conservation Commission and was director of the Wisconsin Geologic and Natural History Society for a number of years. And then he also served as the president of the UW for a stretch when they had an interim need. In addition to those, he was on a number of boards for conservation and had a lot of input into the formation of the USGS and the Wisconsin DNR and Fisheries Commission. So yeah, kind of everything aquatic at that time, Birch was very involved. But his research into lakes centered a lot in northern Wisconsin. He brought his students to the area, and one of those was Chauncey Juday, who was hired by Birch in 1905 and was a lifelong collaborator. Birch was very well known for his mentorship of students, and he started one of the first limnology classes in the US in 1909. That class has grown over time and continues to grow. I think the enrollment now is over 100 individuals. This is a figure that was put together by John Magnuson, who was a former director for the Center for Limnology and a huge part of the history for Trout Lake Station. And this figure shows 1909 and how Juday taught the class for a good 30-year stretch and built the numbers. And this is during World War I. There was a lull in enrollment during World War II, which makes a lot of sense, especially when you see that it was mostly men enrolled in the class at that time. And then Art Hassler took over. Tom Frost taught for a while, and then Jake van der Zanden taught for many years. So this 100-year class has had just a handful of instructors associated with it, but has grown from a few individuals to up to 200 in our high enrollment years. It's gone through things like the Water Pollution Control Act, Earth Day Formation Clean Air Act, and then the Clean Water Act. And so as the course has grown, the topics that have been covered and the placement within a landscape of change has also been occurring. And so just a kind of fun history to reflect on. A couple of features of the Birch and Juday era, and I bring these up because I think they're still very relevant to the questions we're asking today, is that they pioneered instruments and measurements. If you look at the equipment that we use in limnology today, although it looks different, the basic core principles of what we're measuring and how we measure them can still be tied back to some of the machines like this. They're out on the ice looking at gas detection in the waters. Thinking about the integration of physical, chemical, and biological understanding. This device here is from capturing zooplankton at specific depths, and our current Schindler trap looks very comparable to this. And the other thing that was really part of the Juday and Birch era is that they just collected vast amounts of data. They visited over 280 lakes up in northern Wisconsin. And the philosophy that they went by was if we gather enough data, the data will speak for themselves. And so through surveying many of the lakes up here, they developed great understanding for the function of lakes and how that can vary based on landscape characteristics. Not only were those data collected, but those are also archived. So 100 years later, we can reference the original Birch and Juday data. And that's where, as I get into some of the climate discussion, you'll see we have some data that goes back almost 100 years continuously from the lakes that builds on some of the traditional knowledge of the area for us to make some hypotheses and conclusions with. So Birch and Juday had a very, if we collect enough data, we'll be able to answer questions. Art Hassler, we consider him kind of the next generation or the next iteration here at Trout Lake Station, and he was a PhD student with Juday, hired in 1937. During his career, he mentored 53 PhD students and 41 master's students. So a lot of students in a academic career. But he really focused on some new directions. During Hassler's time, he drove experimentation, and this was congruent with the time that he came into his practice, and that people were wanting to look at testable hypotheses and try for experiments. But Hassler looked at the landscape as a place where you could do whole-scale experiments. There's so many lakes, and started developing some ecosystem level experiments where they would manipulate entire lakes. I'll talk about a couple of those coming up. And the other thing that Hassler came into was it was during a time when Aldo Leopold was active throughout the state, and a lot of discussions about how science can really be used to address conservation questions. And so Hassler spent time helping to meld the conservation ethics within the science of limnology, which leads to a lot of the applied research directions that persist today. Along this entire corridor, I didn't want to leave behind, when we talk about all this research part, that we are part of the UW Madison system, and that both at the Hassler Lab and Trout Lake, student training has been a continuous priority. And so this idea of hands-on training that goes beyond the classroom, the formative and transformational part of being in the field with large crews, and that it's community-based. These are all pictures taken at Trout Lake. Our common spot is out on the dock, and you can see an early 1937s crew. This is our group from 1985. A few of these folks are still hanging out today. And then 1996. And just for fun, that was one of the 2 summers that I got to spend here at Trout Lake. And so I got to understand firsthand how formative the relationships, the community, and the training that is received by the folks in the community can be. So Trout Lake Station has had that history and carries that history forward. When I think of the history, we're getting ready to celebrate our 100-year anniversary in a couple of years here. I also think back to the Burge and Judet era, where they visited over 200 lakes with vehicles that looked like this, using trails that the Native Americans shared to access lakes with boats like this. And the reason that Trout Lake formed where it did is because of the railway that was helping with the logging industry during that time. And so Trout Lake Station literally refers to the fact that there was a train station that ran into the peninsula between the North and South Basin on our lake. So getting in a little bit more to the climate and the research directions that have taken place, Trout Lake Station and the Northern Highlands Lakes have hosted a ton of big ideas and internationally influential research. With the Burge and Judet era, they 586 lakes. I undersold it before, but the empirical evidence that they had from that, if you open up a limnology textbook, you'll still see their basic principles in those books. Things like lake stratification. This is something that you've all experienced if you swam in a lake, the idea that the surface waters in the summer will warm up. And then there's a layer called the metalimnion or the thermocline, where it gets really cold really quick, and then the cold layer at the bottom. In the fall, we get overturn. In the spring, the lakes mix again, and then in the winter, we have our ice, and then in the spring, it overturns again before it stratifies. Just this basic principle of stratification, although it seems logical from the outside, there's a lot that goes into that. We have oxygen exchange, temperature change, and the food conditions, the growth conditions in the lake all change during those periods of time. These basic principles come from that early science that was done here at the station. A couple of our whole lake experiments that I wanted to highlight include one called Trophic Cascade that's been ongoing since the time of Hassler, but was really driven by Steve Carpenter in the middle period. And they took a lake up in northern Wisconsin, Peter and Paul Lake, which they divided into 2 basins, and experimentally manipulated Peter Lake. In the Peter and Paul Lake Trophic Cascade experiment, they went in and they took out the fish of one of the basins and left the other one as a control. And they looked at how that influenced the entire food web. When you take out a large predator, what it does is it allows for the lower trophic group to increase in numbers, right? There's nothing eating them. That drives down those little Daphnia and zooplankton that we like to think about, and you can start getting large algal blooms. This is this principle of cascade, kind of the up and then the down and then the up arrows, is one that's been applied across many systems. We hear it applied to marine systems and forest systems, but the experiment in Peter and Paul Lake is one where we actually experimentally demonstrated how the overfishing of some of our large predators in lakes and marine environments can change majorly the algal bloom situations and the overall food web dynamics and health of an ecosystem. In a similar way, we think about if you increase algae, how this can shift in other directions. And so these lakes have been used to test principles of trophic cascade from both the fish perspective and the nutrient perspective in both directions over the last 30 years. Trophic cascade is a general principle of ecology that's been applied throughout the field very broadly. This is just one example of the scale of impact that these whole lake studies can have. Another famous research project that came out of the Trout Lake Center for Limnology group was a study on Little Rock Lake, which, as I sit, is about 8 miles from here, not even, across the road. And it was on lake acidification and acid rain. And in the 1970s, acid rain was a major concern. Many of you probably heard discussions of acid rain for a period of time. At that, during the 1970s and into the 80s, the rain was reading at pH down to 4.7, 4.5. And not to get too technical here, but for folks that think about pH, when we think about pH, a pH of 5, this is a log scale. So if you think about pH, a 1 unit change in pH is actually a 10-fold more acidic, or a 10-fold increase in acidity. So a pH of 4 is really acidic. A pH of 7 is really basic. Lakes around here sit right around 6, and acid rain was falling at 4.5, 4.7. So the rain that was falling on the landscape was almost 100 times more acidic than the water in our lakes.
Could I ask a question?
Yeah, I kind of,
Why was that so acidic, and how was that managed?
yeah. So during, so this was a period of industrialization, and what was happening is that the large roast yards throughout the Sudbury region of Canada and our own processing plants for coal were basically burning. They had what were called open roast yards, where you would just pile logs on top of ore to melt it down, and it would release sulfur compounds into the atmosphere. When they mixed with water, would form sulfuric acid in the upper atmosphere. And so the processing plants would continually release sulfuric acid. And so both roast yards, which were just flat with the surface of the rock and dirt around them, or emissions that didn't put the towers up or have scrubbers. So this was a human-caused acid rain. And in northern Wisconsin and the Adirondacks region and regions throughout Canada, acid rain was a major concern. You would see deforestation for miles and miles around these roast yards. You would see lakes that were shifting in pH. We had statues that were starting to melt, copper statues that would show the signs in DC. And there was a huge amount of talk about the implications for acid rain. So this experiment, and I'll get into our resolution a little bit, Richard, as I go through my story here, because it's one of those where you don't talk about acid rain a lot anymore, do we?
No.
Yeah. So in the 1980s, researchers at Trout Lake Station, Tom Frost, who was a former director here, Tim Kratz and John Magnuson, divided this lake called Little Rock into 2 basins. In one basin, they added sulfuric acid over the course of 6 years to bring the pH of that basin down to the equivalent level that acid rain was occurring. So they brought it down to that 4.7. They maintained it for 2 years, and then they monitored the recovery of that. During this acidification, they kept the reference basin at kind of naturally varying levels. And guess what happened? When you add acid to water, the things in there die and other species become prevalent. We lost most of the common fish species in the water. The algae struggled to grow and different species of algae took over, and it changed the entire community. The other thing that happened, there were some other responses that happened. The fixation of nutrients was more challenging. The formation of shells for snails became challenging as the calcium carbonate got pulled out of the system. And another finding was that mercury became mobilized. It underwent a process called methylation, which allowed for mercury to be released from the shoreline waters at higher rates. And methylmercury is very bioavailable to fishes and can accumulate to create harmful consumption conditions if you were to eat a fish out of a lake like this with high methylmercury. Really what this study reinforced is that if acid rain keeps going and acidifies our lakes, we're going to really struggle to be able to eat fish out of these lakes, for the lakes to be able to persist with the communities that we want to maintain within them. This was one study that clearly demonstrated the substantial negative potential impacts of acid rain. The researchers involved put out over 100 research papers, were invited to present in front of Congress, and this study is one of those that's referenced in the Clean Air Act commonly, and very well known throughout the nation for helping to demonstrate the negative impacts of acid rain. And so, as many of you might know, the response is that scrubbers were required in all, and roast yards were shut down. The cleaning of air before it went into stacks was improved. And then also the stacks had to become much higher in order to move that beyond the, to blow the smoke up beyond the layer in which the sulfuric acid would be produced in high levels. And so really the air that comes out of production plants is substantially cleaner now than it was in the 1970s. Yeah.
That's a great story of remediation, and pleased to hear about it. And I was vaguely aware of it, but it's what I want to ask you is about, and I don't want to get too far out of your field of specialization, but just to give us some perspective, when we talk with climatologists about the planet, they start to get upset about the pH of the ocean, which is now, I think, gone down from 8.2 to 8.1. And they're talking about if it gets down to 7.9, that this would be pretty catastrophic for shellfish. And you're dealing with orders of acidity, at least a logarithm or more acidic than that. And so my question to you is, are oceanic shellfish and other fauna more sensitive than freshwater, or are the climatologists being a little bit over cautious?
So the bulk of that is that shellfish are more sensitive to acidic changes than any other creatures, and corals. So calcium carbonate is, right, that's the white structure that makes the shells of shellfish, or that's why when like a reef dies, that is all calcium carbonate that makes coral reefs or those habitats. And calcium carbonate has a very specific cycle where it transitions from fixable calcium carbonate to bicarbonate to CO2 that can be trapped and released. And the acidity of water is what drives the bicarbonate cycle in our aquatic systems. In lakes, we don't actually have nearly as many shellfish, and definitely not harvested shellfish. The snails in our waters up here show strong impacts of pH, but in terms of shellfish, I think you're looking at crayfish and some of the snails in the surroundings, maybe some ostracods, which are microscopic crustaceans. But in the ocean, the hard-shelled creatures were huge drivers of the evolution of fish jaws, the predatory drive, and the cyclical kind of feedback of predator-prey relationships in oceanic environments and that history of evolution. And so I think the problem within the ocean is if you go too acidic, the bicarbonate never gets fixed into calcium. You can't fix that calcium carbonate. And so you're going to stop losing or stop having the growth of things like coral reefs or the big oyster reefs that you can get. And so there is a tipping point where it just becomes ineffective. And also the softening of those shells is the first step as you lose that calcium carbonate fixation. And so then all of those creatures become vulnerable to predation. And all the big fish and predators have evolved to have jaws that crack the shells they have now, right? This is how it works. It's an arms race. Right now they're winning because they have that material available, but as the acidification would change, that material becomes less available. And so there's both going to be an ecological and a biological tipping point in which it's just no longer effective for those creatures. That's how I think about it.
Well, that's a wonderful review. Thank you so much for that. It amazes me that the ocean is so much more alkaline than fresh water. I never thought about that before. And I guess what you're saying is that the ocean has evolved that way with all of the shellfish that are there, that's released enough lime into the water that it's become more alkaline.
Yeah, it definitely has evolved that way over many, many years. And the evolution of those mollusks and the crustaceans with the hard shells it's a hypothesis that the Cambrian explosion of 540 million years ago, that the expansion and diversification of crustaceans and mollusks is associated with a chemical tipping point that happened in the ocean that allowed for calcium carbonate to be fixed. And so the evolution and diversification of a lot of those hard-shelled creatures is probably tied to some biochemistry and what we're talking about now. And so if you're shifting in the other direction, I think we would expect the loss of some biodiversity. That's how I think about it.
As I gather, some people are kind of fixated or obsessed with corals, and the temperature sensitivity of corals, looking for more temperature tolerant corals, and not spending very much time on the pH requirement, which would affect much more than just the corals.
Yeah.
And particularly the krill and that whole food chain, and that's something we mustn't forget.
Yeah, corals are really sensitive, but they can kind of be used as a first step indicator, but
yes,
they also are huge builders. I mean, Florida's an old coral reef, right? I think we underappreciate how much mass of our shorelines that corals have really contributed over the many years. So when you start thinking about losing them, corals will stick around. It's just that they might disappear for a while, but
yes,
they're going to be there. Just want to show our next few generations them without losing them, I think is.
I'll be talking about the Line Islands in Kiribati and next week, and that's been a wonderful resurgence of corals there. I got some of the information from National Geographic, so that made me particularly sensitive to your thoughts. Thanks very much for explaining that.
Yeah. Lee, did you have a question?
Richard, could I insert a question real quickly, please? Yeah, I just wondered, where do your students typically find employment after they graduate?
Yeah, we have students going every which way, and that makes me really happy. I do think that many of them stay tied to aquatic programs. So the Wisconsin DNR is full of Center for Limnology alums. Obviously, we also have lots of students that move into academic positions at universities throughout the nation. And it's fun because we get to stay connected at meetings and send students to one another nationally and internationally. Water quality. So there's a lot of water quality people out there that are associated with the Center for Limnology. The head of the wastewater management in Madison is a good friend both from Lawrence and has ties to the Center for Limnology. I think USGS, the Fish and Wildlife Service, Great Lakes Indian Fish and Wildlife Service, tribal natural resources throughout the state of Wisconsin and Minnesota. And the other field that I feel is increasing is that we have some students that come to do masters at the Center for Limnology, but then do like an environmental policy. And so they end up being part of the consulting groups, the environmental consulting groups that work in industry. So we've got some that are going into industry. There's fish hatchery folks throughout the state that we have close ties to. So there are jobs. Oh, and the other group is we've been working hard to cultivate a few more science communicators. So hoping that folks can go out and write and present. We've got some WXPR connections, alumni folks that are in those regions as well.
Thank you.
Yeah. All right, other questions while we're at a break point here? I'll leave time for questions after too. I was going to get a little bit into the phenology. Yeah, Louis.
Oh, yeah, I have a couple questions. You have marine background. And is there any way to, what do you think of the idea of engineering phytoplankton to take out more CO2 from the atmosphere? Is that just a really dubious kind of thing? Once you create it, you let the genie out of the bottle and you can't put it back, or is it even viable? I don't know, to have phytoplankton. We always think of tropical rainforest, but the phytoplankton do a lot to pull CO2 out of the atmosphere, don't they?
They can. I'm pausing to formulate my
Yeah, if you choose not to answer it, that's fine.
No, I don't have, I just was scrolling through the nature just put out something about duckweed on lakes and how it's a CO2 contributor, and I kind of read that and thought, what's happening here? I need to read that article. It just posted this morning and I didn't have time to dive into it. I think the exploration of phytoplankton have you seen the contained systems that they're using for phytoplankton as hydrogen, as biofuel production? So phytoplankton grow really fast. And they have them in these coils where they're basically collecting sun, growing phytoplankton, and then trying to harness it for biofuel. So if you could do it in a closed, more agricultural application at a scale that impacted CO2 sequestration out of the atmosphere, I always have trouble if you're gonna change the ocean and put biomodified things into open environments. My background, I'm a farm kid from Wisconsin, and I was a huge, I worked in intellectual property with my dad for WARF during the time of the GMO revolution in ag and corn. My dad's a corn breeder, so I struggle a little bit when we think of putting produced cells and molecules into the ocean environment that are going to change it. So, but sometimes I do consider how that could look in a closed system. That opens up a huge can of discussion, but I think a closed algal system where you're pulling in CO2 from the atmosphere and potentially making a sustainable, harvestable fuel source is an interesting idea.
Yeah.
I don't know if that answers your question, but I'm not ready to farm phytoplankton in the ocean so that we can pull out more CO2. But I'm curious about the discussion.
Yeah, I heard of someone trying to engineer or breed phytoplankton to work at higher temperatures in the Arctic regions. And I don't know if you even like that concept, but since the Arctic is warming a bit. Anyway, we can leave that topic. I just thought, you might have some ideas about it. I have one other. Go ahead.
They are also planting thermally tolerant corals to restore some reefs in the Indo-Pacific. So they're
Oh,
intentionally taking ones from warmer waters and moving them into areas that are warming more. So humans like to experiment with those things, and I'm a cautious person when it comes to redistributing genetics and biotechnology that we build, but I understand the experimentation and interest.
mhm.
It's just hard. Those are hard questions. That gets into a field that I
Yeah,
don't mind
yeah.
discussing, but that is
Well,
not my expertise, just my.
yeah, but you have a background that relates to it for sure. And you can save this question for the end, but you see the current Supreme Court doing anything to unravel some of the work you guys have done down the road or not? You can ignore the question or just address it at the very end or not at all. Because they're starting to get into regulating things. It seems like they're making decisions about things like that the NRC would only do and the EPA would only do, and I just get some bad feelings about where that might go. I don't know. All the work that's been done, like with the Clean Air Act and so forth, I don't know.
I would like to save that one for reflection and a little bit later, if that's all right.
Absolutely. Absolutely. Those are hard.
Yeah.
Okay. Let's move on. Move on, please.
All right,
Sorry
all right.
about those.
No, that's good. I don't mind them. I just, well, yeah. Yeah. All right. So that was some of the longer ago history. I just think it sets the framework for talking about the long data sets that I'll share in the current phenology work that we have going on. And so LTER is called the Long-Term Ecological Research Program. It's been funded by the National Science Foundation since 1981 here at Trout Lake Station. And it was really a brainchild of John Magnuson and other researchers who came together to say, you know what? Big things are changing, and we can't understand it if we don't have some continuous data across the nation that allows us to ask these questions generations down the road. It's incredible foresight on their part, and it's one of the flagships for the National Science Foundation up to today, where so I think we have 28 sites worldwide. That number shifts periodically depending on sites adding and falling off. But Trout Lake Station is one of the oldest sites for the program and has been continuous. We have 40 years of sustained publicly available data. And these data form the backdrop for large-scale experiments and give us long-term perspective that's used for researchers all throughout the globe. And so John Magnuson was the originator of that. He's still active at the CFL and does a lot to archive our history. Steve Carpenter led the LTR program for many years, and Emily Stanley has taken up the reins and is doing a wonderful job of keeping us going and on task. So up here in the Northern Lakes, in 1981, they chose 7 lakes, including Trout Lake, and then 5 lakes around, or 4 additional lakes and 2 bog systems, where every 2 weeks researchers go out and collect information on water quality, zooplankton, phytoplankton, temperature, nutrients, and then every year they do fish and plant surveys. And then in 1994, they added 4 lakes in the Madison area that now are also continually being monitored: Mendota, Monona, Lake Wingra, and Fish Lake. So this is really at the core of a lot of the questions that we are able to ask. And when I talk about climate and phenology, data that we really rely on. So the other cool thing about this data set and John's early work was that he was a visionary winter limnologist. A lot of prior kind of concepts about lakes is that they freeze over and nothing happens underneath, right? It's a period of rest. I think really what that boils down to is it's a period of rest for researchers, because going out, drilling, feet through the ice and collecting data is a huge challenge, was unsafe in past conditions. But we sample all winter long. And trust me when I say winters up here are still very real. They're also very cultural, right? We collect resources through the ice. There's a long history of native fishing through the ice, substantial resource available, but it's also part of the lifestyle and community that we have. People are active throughout the winter, taking advantage of the ice environments. And so winter is a very relevant period, not only for humans as they interact with lakes, but also for the organisms underneath. It's kind of crazy to think that things under the ice go dormant or fall out when I can put a piece of bait down there and the fish are still biting, right? We know they're eating. We know the food web's still cycling, maybe not as fast, and maybe there's some different creatures active, but it still is the period that allows for the spring reset and things to happen. So the seasonality of our lakes in this area is very important. And our current research builds from this long period of discovery. So here's where we get into some of the climate and phenology work. These are the data from ice cover duration on Lake Mendota. It's one of our longer data sets. And what you can see is, so here we're up around 120 days. And so since 1850, the ice duration on Lake Mendota has decreased by almost 30 days. Obviously, there's variability around that. But when you look at the 10 longest ice durations, they occurred prior to 1900. And our 10 shortest durations, the bulk of those are within the last 50 to 60 years. In fact, there's been a couple of years recently when Lake Mendota has almost no ice cover, right? Or no period of ice on Lake Mendota. This pattern is shared here in the North Woods. So in addition to a decrease in the ice duration, one of the patterns that we've seen is a change in our ice off date. And so this is the difference in our ice off date from one year to the next. And you see that in the these are just that 40-year data set. If you go back to the 1980s, you see that you have maybe 5 to 10 day difference. Sorry, this is in days. 5 to 10 days difference in ice off. That's pretty standard. But if you look in the last 10 years, we've had differences up to 50 days in terms of ice off. So 2012 and 2013 are the two I'm going to draw your attention to here. In 2012, the ice went off 52 days before it went off in 2013. So we're talking about mid-March in 2012 and early May in 2013. Last year, the ice went off 30 days earlier than it did this year. And so this interannual variability, and 50 days, you're talking about almost 2 months more of algal growth and zooplankton growth. You're talking about 2 months of walleye spawning and growing before the ice would even come out in the next year. And so organisms that evolved to the stability of these early days are now experiencing really increased variability. And so when we look at variance, I don't want to overdo, but variance in ice off is about 5 times higher in the last 10 years than it has been for the last 100. And that's one of the things that really concerns us, is how do the organisms that were used to these stable environments deal with year-to-year variation that is this high? And I'm not going to give you all the answers today. This is an active area of research where we just began 2 years ago to really dive in, but I will show you some of our early findings from the long-term data and that 2012-2013 change. So we call this phenological whiplash in lakes, and I'm working on this with a group of researchers from the Center for Limnology, Hilary Dugan and Noah Lodig, and DNR researchers Zach Feiner and Greg Sass. And we think of ice off as a critical transition period. It changes the light conditions in a lake, the amount of energy coming with that light coming in, the mixing of the water, oxygen level, and gas exchange. And all of these things have huge implications for physics, chemistry, and biology, all the things living in the lakes. It also has impacts for our research and our access to gathering data. For example, this is the 2012 and 2013 representation. Here in 2012, they thought the ice was still going to be strong because no one ever thinks on March 20th you got to worry about ice up here. And so they managed to get it out, but it really wasn't supposed to be that soft yet. And then in 2013, our students start coming up from Madison, getting ready to get their grad research rolling. And it's May 10th, and the students are arriving later that week. And this is what Trout Lake looked like. Not a great time to send students out to collect research. So, what happens? I talked about stratification earlier, kind of setting this up. When we think about ice going out, what happens? This is 2012, when the ice went out early in 2012. This is the temperature of the lake over the entire course of the summer. So the red is warm and blue is cool. And what you see is this is your lake depth. And so this is from a lake called Sparkling Lake. It's part of that long-term data set. What you see is when the ice goes off early, you have a period of still cool water, but as it warms, that warm water pushes down, and you have warm water all the way down to kind of 12 meters, a very warm core surface area, and this is a huge volume of water that's warmed. Late ice off, 50 days later in 2013, what you see is that the water warms up, but the depth of that warm water column isn't quite as great, and you get a more we call this a more formal stratification, where it's a hard line of temperature change versus warm water reaching all the way kind of to the bottom. Warm temperatures equals energy, right? And so in 2012, this lake is containing a ton of thermal energy. You also lose some of your cold water refuge for fish like walleye and others that require that really cold water for a lot of their life history and behavior. And so 2013, you don't have as much energy going into the lake or stored, and you also have a much larger cold water refuge. These are two extreme years, and neither of these are what the organisms have really adapted to over those long, stable periods of lake stratification. And so again, you see kind of big differences in the temperature environments. This one's a little messy when we look at algal growth in 2012, that warm early year. You see that they have much more biomass of algae, so this is chlorophyll, in that 50-day period, and it's not until kind of early July, mid-July, that the two years merge into just the amount of biomass and algal production that happens within the lake. Zooplankton community, those are the guys that are eating the algae. You see that they also increase more in the early ice off year. And then you don't get until, again, late July, mid to late July, where they kind of match. And so you have kind of a 50-day lag in both the algal production and the zooplankton production in these years. Yeah, Richard.
If I understand correctly, you're finding a biological explanation for the compressed variability in temperatures. Is that right?
I actually think that these biological are responses to the temperatures.
Yes, but I guess what I'm saying is it's not totally random, and it's the biological things responsible for the temperature changes that explain this remarkable compressed variability that you show in your curves, as opposed to temperatures that would stay up for several years and then go down. In other words, instead of a slow variance, you're seeing a rapid variance, and it's based upon the biology of the system.
I don't know if we've got to causal, to be honest. These are the review of the continuous data. I would not say that the biology is causing the temperature cycles. I do think there's feedback loops, but I don't think that I have those connections solidified yet. So I think that right now these are really responses to the temperature increases at different time points in the year is how I'm presenting these right now.
Sure.
But I'm not opposed to feedbacks.
Sure. Okay.
Yeah.
You're being cautious.
Well, I think that the temperature dynamics are driven more by weather. I think it's more that climate change, the weather dynamics, and the El Nino, La Nina, like
Sure.
the global climate
Mhm.
definitely impacts the temperature environments in these ice-off conditions up north.
Okay, thank you.
So taking it up another trophic level, we are always interested in our fishes of northern Wisconsin, and so here we have perch. Perch are interesting because in early ice-off year, you see that the perch populations do pretty well. You get nice big fishes when it's warm early, so that's the red bar here. In late ice-off years, the fish don't grow as big. Makes sense. They don't have as much time to eat in that early growth period. And then in average ice-off years, they do kind of average. And so perch are kind of matching. If they have a short growth season, they're shorter. If they have a mid. And so this is kind of based on temperature. What's different is the walleye. Walleye, we're looking at fish length in really early ice-off year, they're here about 240. In a late ice-off year, they're still really short. But the average ice-off years, the walleye tend to grow larger than either of the other 2 years. And so walleye growth is better in average years than it is in an early ice-off or late ice-off year. Perch growth seems to match with the season. We took this one step further because walleye are really a relevant species within the state. Zach Feiner led a paper with all of us on the walleye, looking at the spawning date of year. And walleye are especially sensitive to ice-off. In some years of late ice-off, they'll actually start spawning underneath the ice. And in years of early ice-off, they'll spawn for over a month. And so their variance in spawning date is also 5 times higher now than it was in the past history. And this figure is a little complicated, but I can try to simplify it. This is the number of walleye that do well. Let's just say this is like how many walleye do a good job. So we want this number to be as high as we can for a healthy walleye population. And if you look, these are deviations from the middle. So all of these light dots are average years for walleye, for temperatures for the walleye. The walleye are doing really well in average ice-off dates. The red and the blue dots here are extreme early or late ice-off years. Again, the red and blue dots that are darker right here are more variable off average ice-off years. And so in these years, when you either have really early or really late ice-off, the walleye spawning is, or the abundances are lower than in these average years where they have the potential to do a lot better. Of course, there's other factors that come into this, but you do see that those most extreme years, the walleye is just not succeeding in their spawning and the subsequent production of offspring. Walleye are declining throughout the Northwoods region, and it's a continual investment of stocking and propagation of our healthy populations. So this could go a long way into advising even stocking investments and how we can best manage some of our walleye populations for the state. So we take this in all kinds of directions. Right now, we've got a lot of questions building off of this phenology framework. And one of them is, as I showed throughout those last few slides, this idea of what's called mismatch or asynchrony in our food web. And so if you have an average ice-off year, the algae comes in, the zooplankton comes in, everybody's fed and happy, and then grows the fish that are commonly found in the lake. This is all optimal timing based on evolutionary history, right? The period of stability of average ice-off. Early ice-off, there's potential for asynchrony because you get algae growing early. Zooplankton require kind of light condition cues in addition to temperature to hatch out and become abundant. And so those can be offset from the algal abundance as it pops up early season, which then can mismatch the abundance of the zooplankton for those spawning fishes that come in and need that resource. Similarly, late ice-off years have the potential for asynchrony or mismatch in that really when the ice goes out, everything starts booming at once and you don't have, again, enough potential mid-level to feed the fishes. These are hypothetical figures. this question. Like I said, we really dug in to try to answer some of these over the last couple years. And our mid-process in this year was a late ice-off. And so we're working up data from this last summer. But these are some of the questions that we're tackling with climate and phenology in our lakes. So take homes, I'm getting close to the end. Climate variability is a growing concern and the implications are real for lake systems. We need long-term data and large-scale projects to tackle these big questions, and the knowledge from these big questions can and should provide guidance for sustaining our waters into the future. And then from the Center for Limnology, just to take it back full circle, we really see ourselves as training the next generation, serving as a source of information, and that it's through the integration of outreach, training, and research that we hope to continue working on our freshwater sustainability. And for fun, just a couple of our outreach opportunities. So we do a Science on Tap every Wednesday that's virtual and live, so it's a hybrid. And this coming Wednesday night will be on Alzheimer's and mental health from the Medical College of Wisconsin, and it's open to the public. And we do our annual open house here on station, and there's one at the Hassler Lab and a number of educational projects with K through 8. And another program that's really booming right now is the Drawing Water program, where we're integrating science and art and trying to reach an additional audience and communicate our science in some different ways. And so I'm happy to answer questions, not only about the science parts, but also about some of the outreach opportunities and interests that folks have. So that's all I have to say formally. Yeah, I see a hand, Steve.
Yes. You have over 100 years of data. Have you done any correlation between the solar changes and the water propagation or other features that may be affected by the insulation being different?
Not personally, right? But the LTR network is, I think there's 15 PIs from the university from different departments working on that. So my answer is that I know of 2 groups that are kind of working on the weather and water relationship. Ankur Desai from UW Madison has done some work on the relationship of weather patterns and these lake phenology dynamics. And I don't know how much solar he has brought into that specifically, but his work would be a place where I would go to look for that, but I don't have a straight answer from his work. The one that I do know better is a researcher called Steve Lowhide, had a graduate student here at Trout Lake. And the link he was making was actually related to solar radiation in association with the trees on the landscape. So he was looking at how trees on the landscape used solar radiation to change evapotranspiration and how that related to the lake levels, which are on kind of a decadal cycle. And it was interesting. He came up to Trout Lake Station to evaluate the impacts of drought on this interaction, and it was the 10 wettest years of history here. And so my real answer is I don't know, but those are 2 places where we might be able to find more information or dive in. And so what we do know is that the trees buffering the lakes are a huge part of regulating how much lake depth changes. And I tie that all back to solar radiation, right? Evapotranspiration is really driven by solar radiation. And so I think that the impacts of solar are probably going to be felt more through the forest than the lake surface itself.
Okay, thank you.
Yeah.
Any other questions out there?
Yeah, I have one question.
Luke, sure.
Yeah, a few years back, Lake Michigan kind of had a lot of ice, and then the levels went up quite a bit. Do you notice that with the lakes that you're dealing with, Gretchen? I mean, if there's in other words, a longer What was the term ice? If the ice off comes late, does the levels go up because they don't evaporate as much? Is there a connection to that?
We don't see that as much in the lakes around here, and it's partly because there's an interaction between snow and ice formation. And so, right, snow is precipitation that then when it sits on top of the lakes limits the formation of what we call black ice downward in the lake. It almost insulates and buffers it. So I think because of that interplay between precipitation and ice formation, we don't attribute lake level specifically to the ice itself, but it's an interesting question. One of the bigger reasons why I think I came from the Mississippi River where I was doing quite a bit of research. And so when I think of the link between snow and ice, the rate at which those melt on the landscape and the ground thaws really can change water levels in systems that have a little bit quicker turnover time. So I think of that river level and how much the melt impacts it, especially in large bodies of water that have a little bit more. So maybe Lake Michigan as a drainage rather than just a standalone lake is how I would think that. Whereas many of our lakes are not as interconnected as some systems for
Instead of
lake level.
Lake Michigan has big open areas, I mean, because of its total area, that might be a big factor. I don't know. Well, thank you for that response. I appreciate it.
Yeah.
I have a little question that's on the periphery of your area. Gretchen, 60 years ago, I used to work with the Canadian Department of Fisheries while a medical student on Lake Huron, and we studied the sea lamprey that came around the Welland Canal, came around the Niagara Falls and got into the Great Lakes. And we were trying shocking techniques and various lampricides and not getting very far. I've heard that since then, the lampreys population has gone down simply by virtue of taking out enough of their own food supply. The lake trout have a fine scale, and the lampreys were preferring to parasitize the lake trout and got the population down low enough that sort of an equilibrium was formed. Do you have any thoughts on that? And lots happened in the 60 years that I don't know about in terms of the lamprey populations and why I think they've gone down somewhat.
Yeah, I think it's not an area that I have a ton of knowledge in. The Great Lakes, my understanding is we still get a little bit of boom and bust cycle that follows kind of those predator-prey dynamics where when you get one going up, the lamprey will come in. I do know that the statewide application of lampricides is still an annual investment. And so there is still a ton of management with the lampricide usage. The USGS over at La Crosse is one of the lead research centers for the lampricides that are being distributed. And so I do know that they're actively applying lampricides to many of the tributaries to the Great Lakes in an effort to keep those numbers at bay. So it is active management to keep them down, in addition to probably some natural cycling, but that's the extent of my knowledge on it.
This is mostly in Michigan State or Wisconsin as much as Michigan?
I think of Wisconsin going up into the Superior watershed is kind of where I was thinking of, and the Mississippi drainage are the two that I'm familiar with.
Sure.
I don't know as much about the state of Michigan, but my guess is they're also doing some lampricide work.
So lampreys are in the Mississippi as well?
Yeah.
Oh my.
Yeah, they're in the drainage, yeah, for sure. So
Oh gosh. Thank you for that update. Appreciate it.
yeah.
Any other questions? I'm checking the
I
chat.
saw Steve with a hand up. Was that Steve's hand?
Yeah,
Okay.
I was wondering if there is any linkage of the work that's being done here in the U.S. to other countries. You know, nobody except maybe Russia has a Lake Superior type environment, but there are a lot of lakes that have come and gone in our heat cycle here.
Yeah, I think that we are in a period of data revolution in terms of sharing data. This is, historically it was hard to collect high frequency data and long-term data. And there was also a bit of ownership and wanting to have that resource held. But the data sharing revolution of the last 10 to 15 years, where all of our NSF collected lakes data goes into a public repository within usually 6 months, if we can get it up there, means that people all over the world are referencing our data and people all over the world are putting up their information. So the majority of, so we've got researchers this week are at what's called the Global Lakes Ecological Observatory Network. It was actually a program that was started by one of my predecessors here at Trout Lake, Tim Kratz. And it is just that. It's international researchers coming together to look at the global lake data, both from these long-term data sets, but also from some of the high-frequency sensors. So now we've got machines that can go into lakes and measure water quality, temperature, oxygen every 10 minutes, and to put those data together for the globe and to ask these questions. And so some of the epicenters of that are in Switzerland and Lake Constance in Germany is a really big part of that. There are Russian researchers and folks from Lake Baikal that are very active. The African Rift Lakes are well represented in those data and comparisons, even though they're not temperate and freezing, they still have dynamics that are affected by these global patterns of weather and temperature. And so there is an entire group that's meeting this week. They meet annually, but they also have a lot of workshops that are asking those at the global scale. And so and the Center for Limnology is very active in that community and one of the hosts for the Great Global Lakes Ecological Observatory Network. It's called GLEON. I can put their link in the chat if that's helpful. But yes, my answer is definitely people working on big lakes, trying to compare across the nation, the globe, wanting to tackle some of these bigger questions.
One of the things that I found interesting on a trip to Iceland was that the fishing in lakes in Iceland is very well managed in terms of the strict licensing you have that is down to the day in some cases, and the number of fish that you take. So I'm wondering if there's anything particular about the Icelandic environment that might stand out in this type of data.
I do know that Iceland is well respected for their lake and fisheries data. And my understanding is there's like 2 or 3 focal lakes that have been really heavily, that have a lot of information from them. But yeah, I don't know the specifics of their dynamic to answer your question too well.
You know, it was very interesting to find out that you had to pay specifically for a lake and for the time period and for the catch.
Yeah.
You know, that would seem like the ideal data.
Yeah, and those are hard to come by, especially in our structures of, freedoms and reportabilities that we've put in place. And so it'd be interesting to see how that could look. I do know that the Wisconsin DNR, we work closely with Greg Sass, who's just about 15 miles from here at the National Highlands Fisheries Research Unit for the Wisconsin DNR. And they have 5 lakes in that region that they do creel monitoring on 365 days a year. And so they have data sets like that from 5 lakes over there, where every fish that's caught is measured and tagged and brought in and out. And they also have tags in most of the fishes, most of the large fish like walleye and muskie, to where they're trying to track some of those dynamics in lakes that are being fished. They also do whole scale experiments where They limited muskie or walleye fishing for the last 3 years, and they just opened it up to see how harvest numbers can be sustainably maintained within a system. So they're doing some whole lake stuff over there, but it sure would be hard to enforce reporting like that for the 3,000 lakes that are throughout just these 2 counties up here. It's an interesting idea, but that'd be a big undertaking.
Yeah, Iceland ideal with the smaller number and, restrictions, smaller population, all sorts of things that are very unique.
Yeah, and residents versus tourists, and we've got our traditional harvest as well. So I think we have, yeah, it's already pretty complex, but it is nice to have the information when we can get it.
Dorothy, I think you have a question.
Thank you very much. It's been very informative.
Great.
Yes, I do have a question. I was wondering if there were any formal ties with any meteorological departments in coordinating research.
Yes, Ankur Desai, who I mentioned earlier, is in Earth and Atmospheric Science, and he drives. We've got, what are they? We've got 4 atmospheric monitoring towers within the Tri-County area right now that are being maintained by Ameriflux, and that data is being managed through Ankur Desai and a couple of our technicians here. And so the other part of that, I mentioned GLEON, the global lakes. We also have the National Ecological Observatory Network on a couple of lakes up near us, and they have additional weather and meteorological data being collected at each of those. And so those data are all available, and Ankur is the one who does a lot of analysis with that. I am a little bit more of an organismal person, so I sometimes reference those data, but haven't dug in as a primary research for my work.
So there's no particular study that is coordinating 2 aspects for how they intersect. So like a combined paper of some sort coming out with researchers integrating their results or not.
Oh yeah, there's a ton.
Okay.
I'm just saying
Okay.
I'm not. I get to help make sure the technicians collect the data properly. Those papers are not ones I'm involved in, but
Okay.
in that network of 15, Emily Stanley and Ankur Desai. Emily's the lake person that oversees the LTR program, and Ankur would be the 2 that would be working on the ecosystem weather global scale. And I know, trying to think, I know they just came out with a paper a couple months ago. They are 2 of the most prolific people that I know. So I would say that if you wanted, I could send you an annual paper with that integration shown. Just I'm so much focused on the ecological food webs that I try to pay attention to those, but I'm not writing firsthand on those. So it is happening. I'm thrilled it's happening, and I think it's going to be happening more with the National Ecological Observatory Network is just starting to put together a Great Lakes user group to help to focus people more on integrating the weather atmospheric stuff into our biological, ecological, to even strengthen that more. But there are people working on it, and I think we all see the need to increase that.
Thank you.
Yeah.
We've had Ankur, but not for a couple of years, speak to us, and it was wonderful work. We should invite him to come again.
Yeah, he's great.
Broaden that. You were also do some marine work, and I know this is not a topic on marine, and it's getting well past 11. But I'd be fascinated if you could just tell us a few words about your. I think you call them ostracods. Is that right?
Mhm.
Could you tell us a wee bit about where you're doing that work and what attracted you to it?
Yeah, so failure is the origin of most graduate programs. I like to tell all my students that start that. So I went to Cornell all gung-ho to work on acid rain and the impacts on the zooplankton in Adirondack lakes. And I got out there and I took samples from 60 different lakes in the sediments looking for these teeny tiny microscopic diapause eggs. And I processed 60 cores and found 4 eggs total. And that's first year of graduate school. So in that failure, I was invited by another faculty to join him in the marine system to look at a group of luminescent ostracods. So ostracods are about 3 millimeters in size. You find ostracods in every water body in the whole globe, terrestrial. Like you can find them in a puddle, you find them in the leaves of bromeliad plants and the wells of pitcher plants. You'll have a species of ostracod that lives there. And the freshwater ones are also found in marine systems, but in the marine system, there's a group that's diversified in a different way that's only marine. And so within that group, one lineage evolved bioluminescence, so the ability to make light. And so they light up for defense. And so when they get eaten by a fish, they just spew bright blue light, the fish spits them out, and they both go swimming off. But in the Caribbean, that luminescence has been usurped for courtship. And so males in the Caribbean do these really complicated light displays to attract females. Some will go up and do these long pulses. Some will go sideways with short pulses. Some will go down with quick flash pulses. So the faculty invited me to come and check out the system that he originally described in the 1980s. And it was all basic natural history. It was, what's the life cycle? What do they eat? Where do we find them? And I said, okay, I'll come do the life cycle, which we all thought would take like a month, right? They're 3 millimeters in size. You think it's going to be a month worth of work, and you'll kind of know. They live for 9 months. So 9 months later, I'm still cultivating these tiny creatures, and totally committed to a degree of investigation, because the system, every place we visit throughout the Caribbean, we find new species. What we've discovered is that this luminescent courtship behavior, which is a little bit like underwater fireflies, has allowed for them to diversify and evolve very quickly in the Caribbean. And so the courtship itself is probably responsible for allowing so many species to arise and coexist on the coral reef habitats. And so since 2001, I've been working in the Caribbean to describe species and to classify, and then we do a lot of genetics and speciation work to ask how these are evolving across systems.
What family are they in?
Family, ostracod is actually the family, Ostracoda, and then the genus and species are kind of specific, but the taxonomy of ostracods is really kind of complicated.
So they're not a bromelid or I mean they're
Myotocopid.
Pardon?
Myotocopid. So the classification goes Ostracoda, Myotocopida, and then within that so the Darwinulids and those other are all in the Podocopid group, which is totally separate.
Okay.
So Myotocopida, and then the group that we work on. But,
okay. Well, thank you. That's amazing.
Yeah. So that's, and I was able to continue doing that work when I was at La Crosse. I would take students down for like a 2-week field marine course.
Yeah.
And so I integrated a lot of course embedded research with students. And it's pretty cool to take a group of students down and say, hey, we just found 2 new species. Let's see if we can get these described and out there. And in this position, I continue to work on it.
Good.
Yeah.
That's wonderful. Any other thoughts, questions coming up? If not, I will, before saying goodbye, I will just say that next Monday, everyone is invited within reasonable driving range to come to Oakwood University Woods, west side Oakwood on Mineral Point Road at our usual time, maybe a few minutes earlier. We're going to test out an OWL system, it's called, which promotes hybrid communication. So we hope to have a table of about 9 to a dozen people, and we will have a online as well as in-person meeting, and it's sort of a trial that PLATO is experimenting with. And so we'll do that. And the subject matter is called Wild Oceans and Forest Floors. And the Wild Oceans part is taken from a book that's based upon a movie that some of you may have seen on Netflix called My Octopus Teacher. And it's a fascinating thing by Craig Ross, I think it is. And it's an hour and a half long. I think to get the main story, you really only need to see the first half of it. The last 30 minutes is kind of the tragedy, as Paul Thompson calls it, the tearjerker part. But the story itself and the beauty of the oceans, I think, is mostly in the first hour. So I invite you, if you have Netflix at home, to read that as your homework before next week. And if you would like to join us, I know you have a lot of other things to do, Gretchen. You're more than welcome. I'd be happy to send you the link. I think you have it already. But it might be fun to talk about oceans and forest floor, and any other people who would like to come are welcome, either directly or online. We'll hope to find this experiment. We've tried it in the past. A hybrid system hasn't worked out very well. We haven't had the right software, but hopefully the OWL system will work. Again, thank you, Gretchen. It's really been very, very pleasant. I've thoroughly enjoyed your talk and your ability to answer wide fielding questions, which this class always comes up with.
It's fun.
We'll say so long to everyone, and have a good week. Happy Halloween this evening, and we'll
watch out for the creeps.
Right. See you all next week.
Thank you, Gretchen.
Thanks for the
Thank you.
opportunity.